Okay, so thanks very much for the invitation, Christian, and for you coming here to listen. So today, as Christian was saying, I'm going to talk about the evolution of recombination. And so among eukaryotes, and most eukaryotes, sex and recombination underlies the evolution of life cycles, of reproductive systems, different sex determination systems, including the differentiation of sexual chromosomes, but also mating behaviors, among many other traits or phenotypes that you might be interested in. There are very few exceptions, and that's this image over here that we think that of these rotifers that have maintained asexuality for millions of years. More specifically, we know that recombination rates scale when we take all eukaryotes with genome size as well as with chromosome number, as shown over here on this right plot. So recombination occurs typically during the first meiotic division, recombination as defined as the uh, as the mixing of homologous chromosomes. And this relationship, even though it's sort of an allometric relationship between recombination rates and genome size and chromosome number, there is significant variation between the different species that have been measured. And indeed, in a few of them, such as in Drosophila pseudobscura, we find that there is genetic variation for recombinations, both within populations and between populations, and that it might be associated with um, adaptation to different environments. So here, what I'm showing is a study from already a few years ago in which inbred lines were measured for the number of crossovers per genome, and the colors of each of these inbred lines show from which population they came from, either from a mountain population or from a desert population, and you see differentiation both between these two populations in the number of crossovers they have, but also within populations. So there is genetic variation for recombination rates, and more so, there is also genetic variation for how recombination rates change across the chromosomes and between individuals. So these are two famous examples, starting here with Drosophila melanogaster. I think this is for the X chromosome, and this is recombination rate over here, in which different genotypes, here represented by different colors of a cross, are highly heterogeneous. So for example, here the yellow genotype recombines much more at this location than all of the other. I'm having trouble with this pointer. Then, yeah, I guess I have fat fingers. But so there is heterogeneity of recombination rates. And here it's showing three stickleback fish in which along the whole genome, you see variation between not only within chromosomes, but between different sexes. So females in particular, or males in particular, seem to recombine much more at the extremes of the chromosomes. This genetic variation implies that there are genetic variants that modify either the number or the position of the crossovers. And so the problem that we're going to address is what maintains this heritability for recombination rates. Okay, and this is a long-standing problem that has been addressed since Darwin's time, if not before, even if we didn't call it the same way, as shown over here by this collection of books. And the basic idea can actually be traced to August Weismann, almost 150 years ago, in which more recombination, more sex and more recombination generates fitness variants, more fitness variants than not having sex and recombination, and therefore adaptation should be facilitated. Okay. So this has been experimentally shown that there is an advantage of sex and recombination when compared to asexual reproduction. So here it's a classical example with Drosophila with fruit flies, Drosophila melanogaster, by Bill Rice and Adam Chippendale, in which they were artificially selecting for a white-eyed mutant and using using genetic tricks, they were able to show that a population that did not recombine, okay, had less artificial selection was less effective than a population that was recombining. So over here, selection was not limited. And here, a more recent study in yeast, in which it was shown that independently of ploidy level, you do have that selection responses or the increase in fitness in a new environment is higher in in sexual populations than asexual populations. But if you noticed here, independently of ploidy, again, having rare or frequent sex has no immediate or no obvious effect. Okay, so the question becomes, but is it also an advantage to having high recombination rates relative to lower recombination rates? And if we understand, if we have a fitness function of that looks like this, so diminishing returns as you increase recombination rates, then you can have an argument that if you have very little recombination rate and you augmented it, there might be a huge fitness benefit. But if you are here already and you augment it by the same extent, then the fitness increase is not that much. Okay. And so that was the introduction. Okay. And so what I thought of doing today is to go briefly first on the theory about the evolution of recombination rates, then introduce C. elegans as a model to study the evolution of recombination rates, and then talk about adaptation under different recombination rate landscapes and how selection can act on modifiers of crossover distribution. Okay. So one can think that there is direct selection on genes that affect crossing over position and number. And the easiest way, and we have numerous examples, that any gene that messes up with meiosis, and we think that crossover is mechanistically, it's primarily um, it exists primarily to facilitate the proper segregation of chromosomes. So if you mess up with this process, maybe you're going to get gametes over here, these 4 gametes, which are aneuploids. So having an extra copy of one chromosome or several chromosomes, or less copies than others. And usually, these gametes are either inviable or upon fertilization have the embryo is not going to make it, okay? So these are direct fitness effects because they depend on the direct effect of having a mutation, a mutant mutant modifier. More interestingly, especially for evolutionary biologists, it's how modifiers of recombination, which by themselves do not have a direct fitness effect, but can become associated with particular combinations of selected alleles. And we call that indirect selection. And the way it works, it's here. This is a cartoon of haploids, so of chromosomes in an haploid sense, in which red means that it's a deleterious mutation. In green, it's a beneficial mutation. And here in violet, you have two alternative forms of a modifier of recombination rates between these two selected loci. Okay? And this dark violet induces recombination or increases recombination between these loci, while these ones don't. And so as you can see, if you recombine these two chromosomes, you'll start to get two beneficial alleles in the same background, while two deleterious alleles in the same background. These will be positively selected. This one will be purged from the population, while it doesn't happen over here. And eventually, with time, you'll have the fixation of this allele of the modifier of recombination, because eventually it promotes the the beneficial alleles to be put on the same background. Okay. Now, this fitness variance or its selection works by creating differences in fitness between these chromosomes. And so I've shown you in cartoon, but now more specifically, it actually what fitness variance stems from is the amount of linkage disequilibrium that naturally exists between selected alleles. And so imagine, again, an haploid model in which you have these two chromosomes over here, the intermediates, which have a combination of deleterious and positive, or deleterious and beneficial alleles. Even if the fitness function over here is linear, okay, negative LD means that there is a preponderance of these genotypes. And what recombination does, like the cartoon in the previous slide, is to split these beneficial alleles from the deleterious alleles, creating more fitness variance, these extreme genotypes, okay? With positive LD, you'll have, for some reason, the preponderance of these genotypes, these extreme genotypes, and what recombination will do is to bring together the beneficial with the deleterious genotypes, so decreasing the fitness variance, okay? So with negative LD, recombination will increase the fitness variance of the population, and so a modifier of recombination rates between the selected alleles will be positively selected with time. While if there is positive LD, it will be negatively selected. Okay. How do you create negative linkage disequilibrium? There are several ways. Perhaps the most common one is the so-called Hill-Robertson effect. All populations are finite. No matter how large a population is, all populations are finite. And so you can imagine that when a new mutation is introduced in the population, you'll be either in positive or in negative linkage disequilibrium with the existing standard genetic variation. That's what's represented over here, okay? Again, beneficial alleles and deleterious alleles. But with time, there will be, and just by chance, there will be, because populations are finite, and together with selection, we'll have selection for beneficial alleles to be fixed, for the deleterious alleles to be purged, so that in the end, positive linkage disequilibrium will dissipate faster than negative linkage disequilibrium. Okay? So you'll end up with a situation in which you have a preponderance of negative linkage disequilibrium. Okay? That's what we call Hugh-Robertson effect. And there is a second way in which you can generate negative linkage disequilibrium, and this is if there is negative epistasis between the selected alleles. So here, fitness, again, against this haploid model, if it's diminishing returns, okay, meaning these intermediate genotypes, the beneficial and the deleterious alleles, when are closer to one of the of the of the of this combination of positive of beneficial alleles, has this hump over there, then recombination, what it does, as I've shown before, would be to generate these extreme genotypes. And because of selection, because of epistatic selection, you will see that there will be a preponderance of these intermediate genotypes. Okay, so this creates negative linkage disequilibrium because there is more of these intermediate genotypes than expected at equilibrium and with no epistasis. Okay, this only happens with negative epistasis, not with positive epistasis. However, with any kind of epistasis, we're actually going to have a different kind of effect, and that's actually manifest already in the offspring, in the first generation upon recombination, which is if we have either negative epistasis or positive epistasis over here, the offspring of crosses between the intermediate, between the extreme genotypes, either very bad or very good genotypes, their offspring will have on average lower fitness. We call that the recombinational load. So it does not favor the evolution of increased recombination. Okay, so this is a summary of the theory that we have. We have direct effects, we have indirect effects, different ways of generating negative LD on the long term, because it's changing the fitness variance of the population. We have negative LD that can be generated by finite population size or negative epistasis. On the short term, we have a negative effect, selection against modifiers, increasing recombination rate because of the recombination load. So one can think that modifiers increasing recombination rates might be favored when they decrease negative linkage disequilibrium, okay? And this might be important when populations start from a situation in which they have low recombination rates and are adapting to a novel environment, while on the other hand, decreasing recombination rates might be favored when they try to avoid the recombinational load, which might be important when populations are already very highly recombining and adapted to their particular environment. Okay. Okay, so we wanted to test some of these ideas, and for that we're using experimental evolution with the nematode C. elegans. And this is a roundworm, okay, soil nematode, not soil nematode, bacteriophages that eat bacteria in rotten fruit or rotten plant stems, etc. And it's characterized by its androdioecious mating system. What this means in the case of C. elegans is that we have hermaphrodites that can undergo self-fertilization, but that can only outcross when they are mated with males. Okay, so the hermaphrodites cannot mate with each other, unlike, for example, most plants, most flowering plants. And local populations, when they are sampled in the wild, we found that they're basically they're highly inbred, they have very poor genetic diversity, and this is mostly due because they predominantly self in, in nature, coupled with linked selection. And what we know of the C. elegans recombination rate landscape is shown over here. So each of these plots is a different chromosome. C. elegans has 6 chromosomes, 5 autosomes, and 1 sex chromosome, okay, the X. And they are characterized, unlike Drosophila, by being allocentric. This means that the centromere proteins are sort of spread out across the chromosome. And if you plot the genetic position, so the number of recombinants that you can find, relative to the physical position, you see very obviously, particularly for the autosomes, that you have these regions, central regions, which were combined less. So the slope of this function is shallower over here in the centers relative to the arm regions, because the slope of this function is steeper, okay? And then you have the tips, which is mostly telomeres, which don't combine. What we know as well is that there is a fixed pattern of 1 crossover per chromosome, okay, at each meiosis, meaning that there is complete crossover interference. Once a position is designated to become a crossover, it somehow interferes with the designation of other crossovers across the chromosome. By chance, when we were starting, there was just a publication that suggested that this gene, called REC1 gene, modified the recombination landscape of C. elegans. And this is seen here. So just as a curiosity, this was a gene that I believe it was the first ever recombination modifier found gene, that not like inversions, for example, in C. elegans back in the 1970s. And it took 30 or 35 years to actually find the identity of this gene. Okay? And that's when we found it in the literature. And so here it's again the same sort of plot, but here on the horizontal, each of these chromosomes. This is the wild type recombination landscape, very much like in the previous slides. And when you're a mutant of this particular gene, you sort of flatten out this landscape, meaning that these central chromosomal regions now recombine more, while the arm regions, they recombine less, okay? And so what we wanted to do was to do several tests about these ideas that I was mentioning at the beginning. And the first question that we ask, is there a direct fitness effect of this mutant of REC1? Okay, how can we do this? So we used a system in which we had a lab-adapted population with plenty of genetic variation, which in turn was the result of hybridization between founder haplotypes that we found mostly in nature, okay, and 140 generations of domestication to particular lab conditions. From this population, we derived an inbred line by self-fertilization for a few generations. We actually derived several of these lines. And then using CRISPR genetic engineering, we mutated just the REC1 gene to have a particular deletion, which we expected to be the loss of function for recombination. Actually, that's how we obtain these maps over here. And we ask the question for a direct fitness effect if we compute exactly the same genetic background across the genome, except for the identity of the REC1 allele, wild type versus mutant, any allele frequency change in time, generation after generation, of the REC mutant can only be due to direct fitness effect. Okay? So we do these competitions between these strains for a few generations. We measure the allele frequency at the REC locus only, okay? And then we estimate the allele frequency change, which is basically a proxy for a selection coefficient, okay? At the REC locus. We've done many of these competitions, and this is sort of the distribution that we find. That is centered, as you can see, on 0. So we do not find any evidence for a cost or a benefit, for that matter, of this of this REC1 mutant. And we've done this both in the domestication environment and in a novel environment, which will become important later on. We find exactly the same results. If we change the sex ratio to have an idea if hermaphroditic meiosis or male meiosis has an impact on this, we also don't find. We might find if we vary the background of the genotypes that we are of the lines that we are competing. But on average, we do not find a direct fitness effect. The next question we ask is there an effect of the REC1 recombination landscape overall on adaptation to a novel environment? And for that, what we did was to integrate this mutant that we generated back into the domesticated population in a fashion that we essentially got two populations which are exactly the same in terms of standing genetic variation across the genome, except in the identity and homozygosity at the REC1 locus. Okay? And then we submitted these populations to experimental evolution, to continued experimental evolution in the domestication environment and in a novel environment. But before that, we compared fitness, as measured also with competition experiments, just after the introgression, okay? And curiously enough, in the domestication environment, we find that there is a slight decrease in fitness, so they become worse. There is actually a recombination load that I'll try to explain, while we don't find any evidence for a fitness difference in this novel environment, which is salt environment. We just put salt in the medium, okay? How do you explain this? Well, we think that this finding reflects the evolution during domestication of gene complexes, in other words, of epistasis, mostly positive epistasis in the sense that positive or negative, but in the sense that good beneficial alleles have been, if not fixed, at least they are predominant in our population. And the evidence, indirect evidence that we have is twofold. So this is plotting linkage disequilibrium as measured by this correlation coefficient, R squared, as a function of genetic distance. So the expected number of recombinants. And these two, and we separate the arms from the centers. So these two big compartments of the chromosomes. And these two, even if we divide it like this, because they are scaled by genetic distance, they should have exactly the same uh, shape, and they don't. In particular, in the centers, we have more linkage disequilibrium, which is sort of indirect evidence that selection somehow during domestication has created these combinations that now are in linkage. Okay, and this is a GWAS that was done in the domestication environment of two fitness traits that we know are fitness traits, size and fertility, in which we found plenty of epistasis, both actually pairwise when we do these these tests that we were talking about, Christian, and when a focal locus, which are these dots over here, interacts with some alleles are interacting with alleles in other chromosomes even, which are these lines over here, okay? So we have indirect evidence, but it looks like this is a recombination load because we're breaking up these gene complexes that were selected during domestication. In salt, we don't find anything because it's a novel environment, okay? So we did the experimental evolution for 40 generations. And our observation here, I'm plotting the gain in fitness or the difference in fitness relative to the ancestral population. So these data points are differences relative to these numbers over here, okay? So if this one started less, it had to evolve more to reach the same fitness level at the end. And in the domestication environment, we do not find a fitness increase to our level of resolution, and we do not find differences when populations have the wild type rec allele or when they have the mutant rec allele, which is perhaps expected because they had adapted to that environment for 140 generations. And here, this mutant did it probably did not it probably recovered the recombinational load, but to our level of resolution, we could not detect it. Okay. More interestingly, we find in the novel environment that the mutant populations show very limited adaptation. Okay, while the wild type populations show increased adaptation. Okay, so they've adapted faster or to a larger extent than the mutant ones. How do we explain this? Oh, I didn't show you before, but now I'm showing you the difference in recombination rates between the mutant uh, allele, REC1 allele, versus the wild type over here. Okay, so 0 means no difference in recombination rates. This is annoying. And that's basically the same graph as before. And what I didn't show you is that this correlates with the numbers of single nucleotide variants that we have in our population, the base ancestral populations. In particular, these arm regions have way more variation, SNP variation, than the central regions, such that if you do the correlation between these two functions, you'll get this. Whenever you have an increase in recombination rate in the mutant, you have less SNP diversity, okay? Interestingly, or not unexpectedly, using this kind of data of GWAS, we can estimate something that we call genome or genomic irritability. Basically, we can sum up the effects that we find during GWAS to estimate how much of the phenotypic variance in fitness traits is explained by the QTLs that we find. And if we partition the genome into those regions where the mutant increases recombination over here, we find that there is less heritability for fertility, for a fitness trait. So it makes sense that during adaptation to a novel environment, you have less fitness variance under a mutant condition, and thus you respond less, okay, than the wild type. Yeah? Can we actually see this happening in real time by doing pool-seq, so at the population genomics level? So this is the analysis Manhattan plot, or two Manhattan plots, in which we have time series, and we just estimate the regular allele frequency change, okay? And this is the significance of directional allele frequency change. And we do find a lot of responses here in this below zero, but there are independent of REC1 allele identity. We find very few responses that are dependent on the mutant, on the allele identity at the REC locus. Actually, this is not a very good statistic, this allele frequency change. It's a better statistic, and there has been some discussion in the literature how best to find what's called linked selection, which here I call interference. Basically, there are several statistics in the literature. Here, we're using the correlation of allele frequency changes with time, okay, between time points for each particular replicate population. And if we again divide the genome into these regions where the mutant increases recombination or it decreases recombination, we do find, as expected, that the correlation of allele frequency change, okay, is way higher when the mutant has decreased recombination. So there is more interference among the alleles that are being selected, okay? Actually, we can see it at the genome-wide level as well, okay? So we do see that at the population genomics level, we have this interference that gets translated into less irritability for fitness, less variance for fitness, and less adaptation to a novel environment, okay, in the mutant recombination landscape. But now the interesting question is, can we say something about the evolution of recombination itself, okay? Not about adaptation as such. And so can we measure indirect selection on this REC1 locus? And the experiment is very simple. We go back to these ancestral populations that we've used to study adaptation, and we just mix them and do now very similar competition experiments as we've done to test direct fitness effects, in which the backgrounds were exactly the same over here, in which no matter how much recombination, how much crossover occurs, there is no effective recombination, while here, where there is plenty of genetic diversity to be sorted out, okay? So if we see and now track the allele frequency changes at the REC, any directionality on these allele frequencies must be due to indirect selection, to its effects that it has on recombining standing genetic variation. And so, again, the same sort of competitions, and now I'm showing this selection coefficient, so the directional allele frequency change. If it's positive, it means that there is selection for the REC1 mutant allele. If it's negative, it's the other way around. In both environments, we now find that the mutant is being positively selected, okay? How do we explain this? you might have noticed that the location of the REC1 was in chromosome 1, sort of on the left side, and it just so happens that it's an oddity relative to the rest, to the other chromosomes, in that nearby it, it actually increases recombination rate. It does not decrease, even though it's like in the arm region, okay? And so there is genetic variation, at least locally, to sort out and to become indirectly associated with this variation, that it recombines. Now, this result is way more obvious if we decrease the population size of the um, of the populations undergoing these competitions. And this, in other words, it's a test that it is indeed negative linkage disequilibrium that is pre-existing or being generated during the experiment, that the mutant allele is going to break, okay? Because the prediction, unlike sort of the intuitive prediction that you decrease population sizes and you increase selection efficiency, actually, this works sort of the other way around, okay? You decrease population sizes, you increase genetic diversity, you increase negative linkage disequilibrium, so the potential for this modifier of recombination to be selected, okay? And so we find that indeed you increase selection when you decrease population size. Okay, so going back to the summary on the theory, we sort of shown somewhat indirectly or more directly several of these mechanisms, okay? And now what I wanted to see is that there is sort of a puzzle that we find. We find that there is selection for the mutant, okay? The indirect selection for the mutant allele, while at the genome-wide level, it sort of prevents adaptation, okay? So there is some conflicting, there is something that doesn't make sense. And so most, if not all of this theory that has been done, it's mostly about the effects that the recombination modifier has on two selected loci. And it's extremely difficult to actually extend that to what we can call polygenic adaptation, in the sense when adaptation depends on many selected loci. And so maybe we can say a little bit more about this. The only way we can say it is actually by performing simulations in the computer. And so we've used them to understand, for example, about a transient hitchhiking of direct mutants with the surrounding selected alleles. So this is shown, again, the same recombination rate differences, the diversity that we have along the first chromosome. And this is the results of simulations in which we're mimicking exactly the experimental conditions. And what these bars show is how long selected alleles that are nearby or far away from the REC mutate, from the REC locus, stay associated with it, okay? And so the darker means that they stay associated with these selected alleles, with the mutant for longer, okay? And you can see that the dark colors only happen sort of for about 3 megabases for a long period. Sometimes there are some associations with the other side of the chromosome between the mutant and the selected alleles, but they are very transient, okay? They don't last for long because with recombination and segregation eventually, basically with other chromosomes, every generation you lose half of these associations, okay? So this is interesting. It means that indirect selection is necessarily very local. We can ask the question, okay, so now in the computer, what if we move the modifier to another position of the genome, okay? And these are the selection coefficients from the computer. If we had moved the rec mutant, so if it is here, the real position, it's selected in favor. But if it's on the other side, it's actually going to be selected negatively. So it's going to be contingent on the genomic position for whatever reason, okay? In this case, in the simulations, it's because we've coded the selected alleles that they are expressed by definition, but you can think that some regions of the genome, the genes are not even expressed, okay? So they do not create any sort of fitness variant. So it will depend on the position. More interestingly, is that any factor that is going to increase linkage disequilibrium between the selected alleles, and that can be external to recombination itself, say the degree of selfing, okay? With selfing, you have an increase in homozygosity in the homozygous classes. So it's going to increase the degree of linkage disequilibrium. And then we can make the prediction that it should increase the size of the genomic region that is going to become associated with the modifier, okay? So that's what's shown over here. Here it's the selection coefficient of the modifier. That it's the fitness in windows, sorry, of the genotypes that are associated with the mutant. So it's local again, that are these. Here it's the same thing, but where we vary the degree of selfing. So we increase linkage disequilibrium, and at least in the computer, we can get genotypes on the other side of the chromosome that are now starting to become associated with the modifier and actually have a negative effect, okay? So on average, if we plot the simulations of selection on the rec mutant versus the degree of selfing, we can actually go from a positive beneficial or a positive positive selection to a change in sign, okay? And this only depends on the selfing rate over here. These are the simulations. We actually did the experiments, okay? And these are other experiments, also these competitions with plenty of standing genetic variation, but where we manipulated the degree of selfing. And at very high selfing, you'll see that the mutant now starts to be negatively selected, okay? While before, here it's sort of neutral, at least in these experiments, but the previous experiments, which were essentially full outcrossing, it was positive, what I had shown, okay? So this puts more complication into the picture, is that sort of the ecology might have a huge impact on, and the reproductive mode might have a huge impact on how indirect selection on modifiers of recombination occurs. So as a summary, heterogeneous recombination rate landscapes decouple indirect selection on modifiers of crossover position from polygenic adaptation. Okay, so there's a lot of fancy words over there. Indirect selection of recombination modifiers should depend on the local genetic architecture of adaptation, including the genomic features such as which genes are being expressed. The size of the selected genomic region depends on factors determining effective recombination, such as reproductive mode, but perhaps also things such as population subdivision or previous history of the population in question. Okay. So where does this all leave us? I think it's actually, these are nice results because they allow us to shift a little bit the attention between generally increased recombination increases adaptation to a novel environment. I think they reveal that we need to be a little bit more precise. And so, for example, this first question that I asked about, is it true that selection among crossover modifiers can maintain irritability for recombination? We don't know. What we know is that whenever we look for it, we find in any natural population plenty of genetic variation for recombination. So meaning that there are many different QTLs that affect recombination. Okay. And it could be that the evolution that occurs at this genomic level is independent of the sort of more organismal level adaptation that we look at. Another question is that, especially when there is population structure and subpopulations are adapting to different environments, do we really have selection for recombination, in particular to decrease recombination rates? One of the ideas is that, for example, if you have inversions, and if these inversions contain locally adapted QTLs, then you can hybridize the whole genome while maintaining local adaptation because these inversions prevent recombination from messing up local adaptation. However, theory that I've done, or Steve Prue has done a few years ago, suggests then when we have modifiers of recombination to decrease recombination between locally adapted populations in direct competition with modifiers to decrease dispersal between the locally adapted populations, and modifiers that diminish the migration load that we have when we have migration between these locally adapted populations, which we call here modifiers of dominance and epistasis. We actually find that the strength of selection for recombination is the weakest among these. Okay, so this could be interesting to test whether or not this is true. And last slide, my acknowledgement. So most of this work was done by Tom Paré. So that's Tom over here. Okay, graduate student, very talented, and in collaboration with Denis Rose, theoretician. And thanks for your attention if you managed to